Hi, welcome to the Law of Fojo podcast. I have with me here today Kevin McKernan. Kevin, thank you for your time. Thanks for joining me. How you doing?
Excellent. Thank you for having me as well. Glad you're taking on this topic because it's long overdue.
My pleasure. I promise I won't ask you anything about Will Smith and Chris Rock. I think that's been extensively saturated right now. So you let me just kind of get into. We want to talk about the PCR test. And like we were talking about before I started recording, a lot of the dialogue right now has to do with assessing the public health response to COVID and what was done, why it was done, whether any of it was effective. And I think we can both agree a lot of it was likely misguided. But one of the root issues, one of the mechanisms used to guide the public health response was the use of the PCR test to diagnose COVID cases. So we'll get into that, but I want to first have you kind of explain what you do and what your background is and your expertise and how it relates to this topic.
Sure. So my background started actually in as an undergrad at Emory. I was working on SSRIs. These are serotonin selective reuptake inhibitors, and that we began DNA sequencing some of those receptors. That got me involved in DNA sequencing at the Whitehead Institute. This is a place where the Human Genome Project took place at MIT. I worked under Eric Lander and ran his research and development team for about 3 or 4 years there. So we built a lot of the robotics pipeline that ran through millions and millions of PCR-like reactions, something called Sanger sequencing. A little bit more complicated than PCR, but still uses thermal cycling and all that fun stuff. And then after that, I started a company called Agincourt Biosciences. This is a company that licensed a lot of the IP from the Human Genome Project at MIT, and then we turned that into a DNA sequencing company and a DNA purification company that was going after HIV detection, a variety of virus detections that Beckman Coulter ultimately came and acquired that company. We had a Skunksworks project in there that was building a new DNA sequencer and didn't really know how to value it because it was such a moonshot of a project that they spun it out during the acquisition. A year later, we got it to work, and Applied Biosystems, who are the owners of the PCR estate, came and acquired it because we had a very novel way of amplifying using PCR in very, very small droplets known as emulsion PCR. And that became the solid sequencer that went to market. It became the second most dominant sequencer in the market next to Illumina. I worked with that company for about 5 years, bringing that to market, and then also bringing forward a semiconductor-based sequencer that also used emulsion PCR. These are very sensitive single molecule PCR devices that do billions of PCRs in parallel. So I got very attuned and attentive to contamination and what things drive PCR fidelity, so that sequencers wouldn't have any error rates to them. When those projects seemed to kind of wrap up, I decided to do something different, and we jumped into the cannabis sequencing space because we saw there was a big opportunity in the fact that the cannabis plant is a pharmaceutical factory that's more or less generic and can be grown in your backyard and makes a lot of compounds that may have an impact in cancer and epilepsy and a variety of diseases. And we were predominantly using these DNA sequencers at the time to go after tumors, like sequencing patients' tumors and figuring out how they were mutated and if we could specifically drug them. We just didn't have very good drugs. And the cannabinoids look very promising there. So I started a company called Medicinal Genomics. That's where I am now. We study and provide picks and shovels to the industry for PCRing pathogens off the plant, for looking at microbes on the plant, for looking at the genetics of the plant. And so I'm actively running PCR every day of my life and have a lot of experience actually inventing different aspects and how to deploy PCR. So I have experience with the patent system. I've been to court over a lot of these patents in the past. So I've experienced a little bit of the intersection of the work that you do in terms of law and an expert witness in a variety of cases on PCR.
Okay, so you use one word and one acronym there a few times, sequencing and PCR. Can you just elaborate on what sequencing actually is for those of us who have no clue what it is?
That's a good point. Yeah. Sure. Yeah. So PCR is a great way of amplifying DNA. It can take a small piece of DNA and make billions of copies of it. In the process of amplifying, though, it may not always amplify one region. It may amplify your target and a couple other off-target regions. And this is where sequencing comes into play, is to look at how on target your PCR reaction is as you go and you sequence all those products. And when you do that, you can see, okay, what percentage of my DNA sequencing is actually from the target I'm anticipating. So sequencing is the act of actually reading every letter. PCR is like the act of just putting a chapter of a book on a Xerox machine and copying the pages kind of carelessly. And sometimes you copy the wrong pages. But sequencing actually goes and reads every letter to make sure, okay, these exact chapters, pages should be in the chapter, and these ones were accidental copying mistakes on the printer. So it's a lot more precise when you get involved with sequencing than PCR.
Okay. What does PCR stand for?
It stands for polymerase chain reaction. And that's the reason for the acronym is that we use a DNA polymerase, which copies DNA in a manner where it chain reacts, where if you feed it the right ingredients, it will make one copy into two, two copies into four, four copies into eight, eight copies into 16, 16 into 32, 64, or so on. It doubles the amount of DNA in every single cycle. So you'll see a lot of these PCR tests are reporting on how many cycles they're using to amplify the DNA. And that's where I think a lot of the controversy comes into play with COVID testing, is that these came to market, no one really had a good idea as to what these cycles numbers were required to actually be a diagnostic answer. They, if you run PCR long enough, it's going to amplify something. It may not be, it's usually not your target if you go beyond 40 cycles of PCR. That's, one way to calculate the magnitude of the amplification is to do 2 to the exponent of the number of cycles, because it's a doubling of every cycle. So if you double 40 times, that's 2 to the 40th. If you do that math, it's a very, very large number. So you can take a single molecule and amplify it trillions of times with PCR. I think the problem is when you get down to these numbers where you're looking for a single molecule in a sample, like from your nose, it's very easy for the reagents you use to target the PCR to find anything it can when you get beyond 40 cycles and amplify it into more or less noise. So you've got to be very judicious and careful, really, when you get past 35 in PCR to prove that you're not amplifying the wrong thing.
Okay, so and that's a good synopsis for it. But let me kind of hone in on a few on pieces of that one by one. So let's start with let's go to the test. Right, you take a sample. That's a nasal swab, a swab from inside your mouth, and then what is it that we're looking for in that sample? Is it viral fragments, genetic material of the virus? What is it that's being looked for or searched for?
We're looking for RNA from the virus, but it's the RNA of the virus is very large. It's 30,000 letters long, and we're really only looking for a couple hundred bases of it as a proxy for the whole thing being there. That's one weakness of what we're doing with PCR. The other complication here is there's hundreds of different PCR kits in the marketplace. So a lot of these discussions about PCR get all tangled up in the fact that not all of them are the same and they do different things. But some of the earliest tests, that came out, it rushed to market so quickly that they were missing some of these controls that we like to see in PCR. One control we like to see in PCR is if you're sampling a human for virus, you should amplify both the human DNA and the RNA for the virus. You should amplify both, because you want to know how much virus was there relative to the human DNA, right? So the reason for this is when you do these nasal swabs, they can vary tremendously. You can imagine if you have a runny nose, you're going to get a lot of sample on that. And if you have a dry nose, you're not going to get any. So the swabbing process, there's been publications showing that the swabbing process can vary by 10 to 13 of these cycle counts alone, if you just look at how much human DNA comes on the swab. That's a very large variance. That's like 1,000 to 10,000-fold variance in how effectively you can swab someone's nose. So if you're not measuring the amount of human cells that it came from, and you're just counting the virus's RNA, you're really looking at something else. You're looking at how effectively you're swabbing someone's nose, not how much virus is there. So it's very important in these PCR tests that you have a numerator and a denominator. You have the number of human cells and the number of virions that are found in those human cells. The first test that came to market out of Germany from Drosten's lab didn't have this internal control where you look at the amount of human DNA that came with the sampling. So you just get a number for the amount of virus RNA that was there, but you really have no way to anchor that to, well, was it a good swab? Was it a bad swab? Because that can swing the signal about 10 CTs, and that can make the difference between you being positive or negative just on how well you sample this. So those first tests were really reckless, and they went to market and probably had a lot of false negatives and a lot of false positives based on the data that we've come through. Now, they've since been replaced with other more accurate PCR assays that don't just target 1 or 2 regions of the virus. They're now targeting 8 regions of the virus, and they have internal controls. And so the PCR process has gotten better over time. But you as a consumer have no idea which test they're using. There's really poor traceability from what might be an emergency use authorization you see on the FDA's website to which test is getting run at the local CVS. You just have to trust that they're all the same, and that's that. You know, anyone who's in the PCR space knows that's ridiculous. The tests all vary tremendously in their sensitivity and specificity.
Are the tests, or most of the tests now that are being used, controlling for human DNA as well as the viral RNA?
I think most of them have, like the ones from Thermo, TaqPath is, and the way that you'll know is if you look at the EUAs, they'll have a primer pair in there. Usually that speaks to a gene known as RNase P. That's a human gene. And they're looking for that to make sure they actually got a human sample, not, you didn't swab your pet or something, right?
Right.
And it also gives you a sense of how effective the swabbing is. So I think that problem has probably been addressed over time. There's a second issue with that plagues all of these tests that was probably not as well appreciated when Drosten put these things out, but became more apparent over time, which is that the virus, since we're looking at only these partial pieces of the virus, we pick up the corpses of old viruses with PCR.
Right.
So when the virus dies, it has a tendency to stick around for 90 days in your body, even though you're not symptomatic anymore. Now, that's something that no amount of improvement really in sensitivity or specificity of the test itself is going to address. There's like analytical false positives where these primers don't do the right thing, and you get aberrant signals. Those things I think they've hammered down quite a bit in the last few years. But what hasn't been addressed in the last couple of years are sort of the clinical false positives. Like, how do we interpret a positive signal in absence of any symptoms and any physician looking at this? I think that's where the real medical crime has gone on, is that they know now that you can be PCR positive for 90 days and only infectious for 9, yet they quarantine people if they're PCR positive without symptoms. That, to me, is a violation of medical ethics. That doesn't really fit with the first do no harm principle. Doesn't sound like it fits with the Nuremberg Code. There's a host of things that the medical profession does not like to blindly test people without clinician review and just quarantine people based on a PCR test, let alone emergency use authorized PCR test that really hasn't gone through the clinical scrutiny of a traditional 510K approval or an FDA approval.
So if I had COVID in January, after 9-10 days, I'm no longer considered infectious or contagious. I could still technically test positive for COVID if I took a PCR test 30 days later.
You could today, but now here's the interesting thing. We wrote about this in we wrote a paper. There was one of the earliest PCR papers that came out that kickstarted all this was this Corman-Drosten paper. And it got raced through peer review in like 24 hours. The authors on the paper were on the editorial board of the journal, and they had investments in testing companies. So there's conflict alarms everywhere in this, but it gets slammed through peer review. They've never put those peer reviews public, so we decided to write our own and put it public. One of the main critiques we spelled out in that review was that this exact problem is you're not giving us a really valid CT score for this test, and we don't know how to interpret it clinically. It's just a signal of RNA. What if this thing sticks around for a long time? And doesn't have any, and is no longer transmissible, but you're still getting positivity. That's turned out to be the case, and the CDC years later has now put a moratorium on doing PCR on someone just like you. You're 30 days. If you're 90 days out from being positive, they don't want you PCR testing again because they know you'll still be positive.
Right. And
you're likely to just, fill up the entire PCR pipeline, because when you test positive, that triggers all of your family members and contacts to go get tested. It creates this kind of chain reaction of contact tracing of overly testing the population. In that process of testing your family members, you're probably going to also pick up 90% of them that will have had it in the past and are no longer infectious because they're under the same 90-day problem that you're under. So it creates a tremendous chain reaction of the number of people that are positive that aren't necessarily infectious.
Is there any way to determine if there's, or is there any way to cross-reference, and I might be using the wrong terminology, but a minimum infectious dose when looking at these test results to determine, does this individual have a minimum number, a minimum amount of viral load or viral RNA to trigger a diagnosis?
So there have been a few ways to triangulate on that. There's a paper from Rita Jaffar's lab, who was working with Didier Riault, the hydroxychloroquine lab in France that treated a lot of patients. And what they did is they took about 3,700 or so patients and they PCR'd them, measured the CT score that they got, and then they also plated those patients' samples to see if they created any plaques. It's known as a plaque-forming unit. When virions are plated onto a certain type of lawn of cells, they'll create death spots in that lawn that are reflective of the number of virions that were plated. So you count colonies, or the inverse of colonies, really known as plaques. Okay. And that PFU gives you an estimate of how many virions that you have that are capable of killing a cell. Not how many RNA fragments, but how many really live infectious virions do you have. And when they do that, they came to a calculation that something like only 3% of the samples after a CT of 35 were actually creating PFUs. So they made a call of don't call anything after 35 because it's no longer infectious. That's one way to do it, but that was on their test, and arguably you'd have to redo that test calibration on every other test in the marketplace because it's unlikely to be 35 for all hundreds of the tests that are out there. It might drift to 33 or 37 depending on the sensitivity of the particular test. But the other angle we have on this is people have been looking at rat tests, these rapid antigen tests.
Right.
And those only trigger on a protein that the actual virus makes. All right, so that's a sign that it's not just DNA, but you've got DNA in the protein coat that's coating the virion, and those seem to have a much narrower window of positivity on a given patient. All right, so they don't have this long tail that PCR has of being positive for 90 days. They also don't come up on day one like PCR does. So PCR has this sensitivity to pick things up a little earlier than the rat tests, but the rat test, once you are positive, the rat test is a better test to estimate when you're no longer infectious. So they use PCR to say, okay, we don't know whether you had this, whether this started yesterday or 90 days ago, but we know you have it.
Right.
If you start getting symptoms, start taking rat tests until you don't have symptoms anymore. And then when you don't, when the rat tests are negative, you should be no longer infectious and free to go about your day. Now, we didn't have that type of policy in place in the beginning of the pandemic, probably when it mattered most, when we were quarantining everybody, including nurses. So this has only really recently become in vogue as they've been sending some of these free test kits out all over the place. But so they're getting better at this. But I think the biggest hole in all of this is that they're still trying to get all this done without clinician review. And many of the COVID symptoms aren't very specific. And they're trying to rely on just these tests to make calls on quarantining people. And they really should have a physician involved looking at the host of symptoms that you have and making a real, a clinical judgment as to whether or not you're in fact infectious. Now, some say that just doesn't scale, but I'd be, if you actually dial back all the positives, I think the problem we have now is we get so many positives with the PCR pipeline that it doesn't scale either. You flood the hospitals with all these people that are asymptomatic or had it 90 days ago.
Yeah.
And it ends up clogging up the whole system from another perspective.
Right. Right. Yeah. I mean, you would think that this notion of scaling, I mean, to me, that's a business term. How do you take a process or some kind of mechanism and then you scale it for growth, for profit? But it seems like that concept shouldn't even come into play here. You're talking about individuals, and a diagnosis should depend not just on one factor like a PCR test, but it should depend on a thorough, a complete review of not only the test, but symptoms and any other complaints or information or intel you can get from the patient, right?
It's very important. And if they're really going to stick with this model of there's nothing to treat this we can only rely on the vaccine, and you have symptoms that are flu-like and you don't know which ones they are, well, the vaccine is going to help your flu.
Right.
And if you offer it, and there are some treatments for flu, right? And they're demanding there aren't any real treatments for COVID other than vaccines and maybe remdesivir when it's too late. So yeah, getting it right is very, very important on knowing how to properly treat the patient and treat them early. I think the problem we have now is we're doing all this PCR and then telling people stay in your house until you turn blue and then come to the hospital when we have a much lower likelihood of ever fixing this.
Right. You mentioned, so you were, in your explanation, you mentioned CT score, amplification a little bit earlier. Amplification, that's the process through which you create copies of the viral RNA that you obtain through the sample. Is that right?
Yes.
Okay. And it is when you mentioned Xerox copies, and I'm thinking, back when I was in grade school, where you have a teacher that uses the same test over and over every year, and he or she makes 20, 30 copies of the test, hands out the test, and I'm looking at this Xerox copy of the test, and it's a very, very poor copy, clearly the result of her having taken a copy of a copy and just doing that every single year. Is that an accurate analogy to describe what's going on when you take a DNA sample like this and just run it through all these cycles to amplify it in order to try and diagnose whether or not this person has COVID?
It is, particularly when you get out to measuring like single molecules, when the virion load is so low, that copying process, what we have in PCR is we're kind of measuring like the size of the stack and the number of papers that when you run it through the Xerox machine, if you had one slice versus 10,000 slices, but you're not actually reading them. There is a probe in there that's about 25 bases long that tells you that, okay, you've roughly copied the right page because there's this one sentence fragment in there that we see every time that we copy this. So we're always checking to make sure at least that sentence fragment is there every time we make a replication of this. That's how the PCR monitors the progress of the amplification. Is every single page we copy, we check if there's a particular word or two that's on there that should be on that page. However, that can be fooled. For one, when you get to these really dilute quantities, you can start amplifying potentially other SARS coronaviruses that may amplify. This is particularly true when you get past like 35 cycles. They don't have, it's very expensive to sequence verify samples that are that dilute. All right, so you'll tend to see in the marketplace people when they do sequence verification of PCR, they're usually only doing maybe 1 to 10% of the samples that go through the facility. They'll graduate for sequencing, but they need the sample to be below a CT of 32 for them to sequence it. It's not to say that you can't sequence the stuff that's out past 35. It's just extraordinarily expensive and hard, and no one does it. The CDC has the number backed all the way out to 28. If you're 28 or lower, they'll sequence it to verify it, and they don't do this in every sample. But that's really what's giving us the confidence that those numbers below 32 are trustworthy, because they have some sequence that backs up the fact that the PCR assays are in fact triggering on samples that have full genomes of the virus. All right, that's like ironclad, rock solid information. They have full genomes of these things. When you get past the later, they don't do the sequencing, so we don't really know. And there have been a couple round robins that have been done where people send flu samples, rhinovirus samples, other viruses through the PCR testing labs. There's one in Australia I can link your readers to, where they test the COVID assays on all these other viruses, and they occasionally light up at 38 or 39 on the wrong virus. All right, so that's the sign of your analogy where you went haywire copying this thing and lost track of the actual target, and you got some pages in there from the wrong chapter, if you will. So it gets very hairy when you get out past 35.
Right. When you say the sequencing is too expensive, is it because it takes too long, or why is it so expensive?
Well, there is a time component. Sequencing usually takes in another couple of days, if not weeks, depending on the method you're using. And when the samples are that dilute, and you're swabbing your nose, you have to realize that your nose comes with a whole microbiome. And when you go about trying to sequence that, you have a couple choices. You either sequence everything, where the majority of it is human DNA and human RNA and a microbiome, and your needle in a haystack might show up. That usually they don't do. It's too expensive because you waste all your time sequencing this microbiome. So instead what they do is they amplify the entire virus genome, not just 200 bases of it, with usually around 500 base pair segments. This is one protocol is known as the Arctic protocol that does this. So they amplify 80 of these different amplicons and sequence them all. And that process is one, you've got to go do that amplification step, and then to purify it and then sequence it. So it's usually takes another week for them to go sequence verify these. So it's not really a diagnostic tool that can work in real time with trying to turn around if you were turning around things a week later, there's no point in track and trace. It's too late. Right. So you have to have something that kind of turns around in a day or two, which is what the PCR pipeline's meant to do, is to just kind of triage the population for who might be infectious. But unfortunately, it's not doing a very good job at discerning the infectious from the non-infectious.
What determines how many cycles to run on a sample?
So they typically set that at 40. That's kind of the default you find on most PCR instruments.
Is that where does that default number come from? Who
manufactured the instrument? And I think just in general, people have recognized that you usually can pick up a single copy of an organism below 40. And if your test doesn't do that, it's limping along, and there's usually something wrong with the efficiency of the test. Now, some people push them out to 45 because their test is for some reason hindered a little bit and less efficient, and so they want to run the camera out 5 more cycles to see any late CTs. Now, people often conflate this on social media and other places. When they see 40 cycles in the EUAs, they think they're calling people out at 40. They're not. They're running their camera out to 40 so they can confidently call samples at 37. So oftentimes you'll see the EUAs have 40 and 45. That's not necessarily red herring that the test is bad. I'm very suspect of ones that need 45 cycles to figure this out. You should do that. But they're probably calling those at 37 or 38. The real problem is there's no transparency on what people are calling these at. I've tried to ask labs in Boston and others, like, put your CT values public for the world to scrutinize. Yeah, they won't do it. So there is a little bit of a cover-up going on here on what are the CT scores you're using to drive these pandemics on people, because I do think the pandemic numbers we keep getting shown on these waves are grossly exaggerated because you're mostly chasing ghosts. You're mostly chasing these people that had it. I mean, you tend to see these big spikes after vacations, right? Like January, huge spike in January. Everyone went home, holidays, mixed, may have got it asymptomatically, and it's in the rearview mirror. They come back to school in mid-January and get tested, they're positive. They're not actively sick at that time or infectious. So that type of testing, unfortunately, drives a whole lot of panic, and I don't think is necessarily productive.
At what point in the number of cycles does the possibility of a false positive come into play?
So great question. There's 2 different types of false positives I like to itemize. Are these analytical ones where your assay might amplify the wrong thing. I think those are out past 35. There are also these clinical false positives that I think all the tests suffer from, which is you can't discern infectious from non-infectious people. And that, I would argue, is probably anywhere between, for probably 50 to 90% of the samples end up in that bucket. So if you take the CDC moratorium, for example, 90-day moratorium, you know you're infectious for 9. All right, so that's like 10% of the people that you test are infectious, and 90% of them are non-infectious. That would be a 90% clinical false positive rate because you can't discern infectiousness, right? Maybe in some cases that 90-day PCR moratorium that CDC has is a little aggressive. Like some might say, not that many people are really positive that long. It's a real minority. Fine, cut it in half. Call it 45 days and 9 days. You're still in a scenario where for every one person you find that's infectious, you're quarantining 5 others.
Right.
Like when has that been an appropriate legal justification, right? I mean, isn't there like a Blackstone's law? I mean, it's not a law, but it's like a theorem of like, I'd rather have 10 guilty people go free than lock up 1 innocent person.
It's an issue of tailoring. Yeah, it's an issue of tailoring the policy to the interest you're trying to protect. And is quarantining 4 people who are not infectious for every 1 person who is infectious, is that an appropriately tailored policy or action, and I don't think it is. I think you're certainly overshooting by a significant degree.
Well, particularly when the policy's justification is to monomaniacally focus on preserving the healthcare system, and you're quarantining healthcare people in the process.
Right.
So this is the rationale behind it. One, I don't believe the healthcare system is overwhelmed. And two, if it were, why haven't you built any more hospitals? And three, why can't you build more hospitals? Because you have laws in place that prevent it, according to the Certificate of Need law. None of those have been reversed during COVID. So the whole thing's built on a whole foundation of lies.
Right.
So their justification is don't overwhelm the healthcare system. Well, you're PCRing nurses too, and doctors, and they're getting quarantined inappropriately. And most of those people are probably already naturally immune because they've been surrounded by this for 2 years.
Right. So I read a quote from Dr. Fauci at some point. I think it was either in 2020 or late 2020, where he explained that the PCR test is useless and misleading when run at 35 cycles or higher. Are you familiar with that quote? Is that accurate?
It is. It's very accurate, and he knew it before the pandemic started and didn't say anything about it until halfway through, and all the damage was done.
What can you give me some idea, if you know, what does the false positivity rate look like as you increase the number of cycles? So if you're at 35 and it's considered misleading, anything above that's considered misleading, what is the false positivity rate at that point? And if you go up to 40, 45, what does it look like there?
So the Rita Jafar's paper, and folks can Google this, JAAFAR, had 97% of them as false positives after 35.
Wow.
All right, and that was by measuring these things with PFUs on Vero cells. And they did, the interesting thing is they did it on Vero cells, and Vero cells, according to the CDC, are like log scales more infectious than human cells. They did it on a very conservative platform. They measure at the CDC did this study, I think it was called Harcourt et al., where they tried to infect a variety of different human cell lines, including Veros, which are monkey kidney cells. And they found the monkey kidney cells were like 100-fold more infective than the human cells. And so Rita Jafar took that and said, okay, well, let's do this on the most infective cells, be sort of conservative. And even there, after 35, 97% of the samples didn't culture. So that tells you that those people just have dead virus that's floating around, and they're no longer infectious.
Now, was that study based solely on the cycle thresholds, or was it also based on the fact that many people many people continue to have viral fragments in them after they're contagious or infectious?
So that's a good question. That was done just on CT scores, plating. But you're asking a pertinent question I can't recall right now, which is what was the inclusion criteria for the study? I think these were people that were coming into their hospital, so they were probably symptomatic.
I see. Okay. So
the story might actually get more perverse if you include asymptomatic people that aren't seeking out healthcare.
Right. That's right. That's where I was going with that. I mean, if we're talking 97% false positivity rate when the CT score is 35 or higher, potentially could be even higher when you consider if someone who's not symptomatic and who just decides to volunteer for a PCR test, but they're no longer infectious.
It's rarely a volunteered event.
Yeah, I know. Yeah. That's the other issue with this test. It fails to capture people who either had COVID and recovered and never knew about it, or people who just choose not to go get tested. Like I'm one of those people right here. I had COVID, I didn't even know about it, and so I was never captured by this testing process. And I guess you would think that it probably pushed the fatality rate a lot higher when you're missing all these people, right?
Well, there's that problem. Likewise, in the hospitals, there's a tremendous amount of everyone's getting tested multiple times in the hospitals. So the numbers get inflated. Just they're not discounting for multiple tests in a lot of the public databases. So you see these escalations because once you test once, you start testing yourself every third day or so to figure out what's going on with the course of your disease. Those will add to the tally. And this trickles down into people who have comorbidities that might be dying for other reasons, but they're PCR positive as their immune system starts to decay in their final days of life.
Mm.
And there's all these financial motivations for people to bill things as COVID, because there's different reimbursement policies for COVID. There is different liability for COVID patients. So there's a tremendous amount of incentive for the hospitals to get everyone under the COVID umbrella that they can. So they're going to test everyone all the time.
Yeah. Yeah. No, I know. I've read up a lot on that. It's extremely perverse, creates some significantly perverse incentives. If the false positivity rate is 97% when you're looking at 35 cycles or higher, I mean, I checked this morning, in Massachusetts, there have been 1.7 million COVID cases to date, diagnosed COVID cases to date. So if I applied that 97% false positivity rate to, let's say, half of those cases, I mean, because I don't know how many. Well, let me ask you this: do we know how many labs are using the PCR test? I mean, how predominantly is it being used?
It's being used extensively in Massachusetts. In fact, one of the largest testing sites in the country is at the Broad Institute, and they test. If you go to their website, you'll see they have a tally up there. They've done, I think, over 22 million tests. So a tremendous amount of volume going through there. Now, what's not clear is what CT or what percentage of people are 35 or above in their data set.
Right.
I mean, they may not even call above 35. We don't know what their CT policy is for calling these things.
Right.
So I don't know how much we can just, blanket apply 97% false positive across all of them. They may have discounted for a lot of that.
That's a valid point.
In who they call. But I do think it's worth pointing out there's a lot of financial motivation going on in here. If you're not familiar with the PCR industry, usually these PCR tests in a research setting are a couple dollars. In a clinical setting like a CLIA lab, I used to run a CLIA lab that used PCR, and in CLIA labs, you have to have physicians sign off and everything, and there's added cost as a result of this, and the numbers usually get reimbursed maybe $30 to $40 for a PCR test by an insurance company. People now, since there is an artificial demand, I wouldn't call this a real demand because it's being mandated, the supply chain hasn't necessarily caught up. We're seeing testing running for $150 to $300. Now, when that happens, now you may not see this because the price isn't always apparent to the consumer. There are free test sites out there, but someone's paying for that. And they're usually, if you go and pay out of pocket at these places, you're looking at 150 to 300, depending on how fast you get the results back. Okay. So that's a 10x markup, 5 to 10x markup over traditional PCR testing. And what that does to the marketplace is all the other diagnostic tests for more serious disease like cancer, those people can't get reagents right now because they're all getting soaked into the COVID pipeline because COVID has a higher payback for people. Now, you have to think about what's going on globally here. This means we're spending all of our PCR dollars at an extraordinarily exaggerated price, chasing kids that don't get sick for COVID at the cost of testing cancer patients. We've created just a nasty dynamic in the marketplace with government intervention. And now there's supply chain problems where all these boats are off of LA, more supply chain problems with petroleum issues. A lot of the DNA synthesis tools that we use rely on acetonitrile to actually make oligos synthesis, to make the vaccines and to make the PCR reagents. That's hard to find these days. So all the oligo shops are now backed up trying to supply this monomaniacal focus on a disease that's relatively harmless at the cost of us having the capacity to test everything else. And so that's something people need to keep in mind when you're testing their kids. Your kids aren't going to get sick. They're not going to get hospitalized. You're just doing this to placate nervous Nellies at schools. Yeah. It's really not changing the safety profile of what's going on in your schools. In fact, some of the interventions are probably harmful. But behind the scenes, what you're doing is you're cutting in front of cancer patients. And that's never spoken about enough.
Yeah, no, that's a really good point. I didn't know that, and that's it's very concerning. I mean, I've been saying this for a while. This ends if we just stop testing, especially testing people who don't need to be tested. It's like children. Children are obviously not at risk of this of this virus. Right.
I mean, they should stick to symptomatic people at risk, with physicians involved to actually triage these fairly vague descriptors of, I mean, a fever can be from a lot of things, right? Sure. Many of the symptoms of COVID are overlap with those of other respiratory diseases. And so you need somebody a little bit more looking at the whole picture than just a single PCR test. Never before in medical history have we been making medical decisions without physicians on a single PCR test.
It's incredible. Why did we, do you know why we defaulted to using the PCR test to try to determine if someone has COVID?
I think that is a result of the shenanigans and fraud that happened at Eurosurveillance. So this is the journal that those authors submitted their paper to, that they were the editors of.
Right.
And they had money in testing companies. Two of the authors had money in testing companies. So they did this, and now before it got through peer review, it ended up on the WHO's website, despite the fact that China already had a better test published. And it turns out Marion Koopmans was on the WHO committee. So she's probably shepherded into the WHO's website so her buddy could get her test into the marketplace ahead of everybody else. And that peer review was a train wreck. We've written about 60 pages of all the holes and problems in that test. Now, that doesn't mean all the other tests out there are broken. They've filled in a lot of the problems of that rush test. But once that got going, there became a financial motivation for everyone else to replicate this all over the world so they could have multi-billion-dollar testing markets. And that's what we're seeing here in the States. There's at least, I think, Thermo's probably, if you add up Thermo, LabCorp, and Roche, and the numbers are in the tens of billions for testing per year.
Right. It seems like those perverse incentives have kind of driven a lot of what's been done. Everything from hospitals relying on testing everyone that comes in order to get those dollars, those federal dollars. The vaccine industry now. It seems like all of this is being driven by these monetary incentives rather than what's right for the patient, which is, it seems to be, that should be the optimal concern in the medical field when you're dealing with a virus like this or any other illness.
I think this is what happens whenever you depart from personalized medicine and you end up in herd medicine, you invite this type of eugenics crap to the table.
Yeah,
because that's what this is they're looking at the whole population as a herd, thinking how can they optimize herd health while making the most money possible. And so they put in these sort of one-size-fits-all rules that don't make sense. In what New York needs to do is going to be very different than what like Alaska needs to do, right?
Right.
And so it's simpler, I think, for these people to lobby. Once you get this testing train going, these people have tremendous amount of lobbying dollars to then continue and push the teachers unions and push the schools and push everyone into this degree of panic. They can then start advertising and altering the news and the media so that people are constantly in this cortisone shock of fear over this thing. So it has gone completely off the rails. And I no longer trust anything I see in the news. I wasn't this cynical 2 years ago, but now almost anything I see in the news about COVID, I pretty much assume the exact opposite of that is probably true.
There's, going back to the cutoffs for the CT thresholds. There was a I was trying to share my screen, but I can't do it here. There was a document that someone I was working with a little over a year ago obtained from the New Hampshire state government, and they got the cutoff CT values for the public health lab in New Hampshire. And they were relying on the manufacturer recommended cutoff CT values. There were 3 different manufacturers, and the recommended cutoff values were 37, 40, and 40. So does that mean that they were running 37 cycles and then 40 cycles and 40 cycles for the tests that they were
that probably implies there were 3 different labs that were doing the bulk of the testing, and they each had different numbers, and those all sound very high to me. And I'd be surprised if any of them. I'm not familiar with any paper that's come out of a New Hampshire lab that did what Rita Jafar did and calibrated the test against infectiousness or living virus. So most of these people just take anything that's gone through an EUA where the manufacturer sets the number at 37 or 40 and doesn't really go back. And they don't do this necessarily with patients. They do this with test samples. Yeah. And they probably just stuck to that and ran and never questioned the actual CT score beyond that. So those are probably calling a lot of false positives.
Right. Now, when it says that's a cutoff value, are they stopping at 37 cycles? Are they stopping before, but they don't go as high as they don't go any further than 37? Like, where
When
do
I
is
hear
it?
when I hear cutoff value, I think
Mhm.
that's their call value. Mainly because most of these cyclers will default to doing 40 cycles, they'll run the camera for at least 40 cycles. And that's
Right.
not crime, because in order for you to see the signal at 37, you got to run the camera a few more cycles to see the growth of the curve.
I see.
If you actually see these PCR, the data from these PCR systems, they're tracking the growth rate of that Xerox machine over time. And so
Yeah.
they actually get imaging every single cycle to measure the RFUs. So to measure a sample that's kind of crossing the baseline at 37, you've got to get a couple more data points after it to see that curve grow.
I
So
see.
40, running the machine out to 40 is not a sin. But when I hear people say cutoff value, I think that's probably their call value, that they're saying
Mhm.
anything that has a CT value less than this, we call positive. And those are all very high numbers. 37 and 40s are very high numbers for this.
Yeah.
I'd be
So
surprised if they have any sequence validation to back up what they're doing. It's hard to sequence stuff that's out at 37.
right. So it's I think you mentioned just like a minute ago, it's safe to say that there were a lot of false positives coming out of that lab.
I would, yes, I would assume so.
Yeah. That's incredible. I mean, if especially if you take these high percentages of false positivity rates and apply them to, like I mentioned before, 1.7 million COVID cases to date in Massachusetts. Let's just say half of those were false. I mean, that is a significant number of false positives.
If you want to call it, clinical versus analytical, I bet
Hmm.
clinically more than half of them are false, because they're people who have it in the rearview mirror.
Yeah.
Now, if you get down to the bucket of people who are in fact infectious, there's
Right.
still an analytical false positive rate amongst those, which is, depending on where the CT is cut off, it could be, as high as 97% if they're calling everything after 33 or 35. I doubt the bulk of the data is beyond 35. When I see a lot of this data, it's usually between 20 and 35. But if you have labs that are calling them out at 40, there's going to be some percentage of people that are out at that range that are just probably analytical false positives. So, and even amongst the ones that, the point I want to get across is there's how accurate the assay is an analytical question, and
Yeah.
we can dissect that as much as we want and and that's very test dependent. So
Yeah.
those arguments go in circles. Someone, the moment you say that test is bad, they bring in a different test and say, well, check this one out, right?
Right.
The reality is all of the tests have this problem of not being able to discern infectiousness versus non-infectiousness. And I think that puts about half to 90% of the people as being non-infectious when they're positive with PCR.
Yeah.
So yeah, take the 1.7 number and assume at least half, if not more, were non-infectious people that were asked to be quarantined. And then you see what happens to society. It just wrecks the economy.
Yeah, I mean, that's 850,000 tests that are likely false positives. I mean, and you're driving a public health response based on
You're
these.
shutting down businesses, you're shutting down schools. The impact on this in terms of like starvation in other countries, like third world countries, our supply chain is not making it to them. So this is a real privileged position to be in, which is to say we're going to lock down the world. It's a laptop class privilege, frankly, because they're not paying any attention to the fact that when you arrest the economy like this, you may not feel it in your ivory tower, but the people in Africa are starving because you are a hypochondriac.
Yeah. What was this test invented for? What was it used for? And what was it intended for? It's clearly not a good diagnostic tool, but so what's its use? What is it useful for?
It's used. It actually is used a lot in diagnostics, but usually with multiple other data points, so that the doctor can discern what's really going on. And the reason for that is that when you look at things at the DNA and RNA level, you're a couple steps before disease, right? DNA gets turned into RNA, that gets turned into protein, and that sometimes has a medical effect or changes the biology of the cell. When you're measuring all the way back at the DNA and RNA level, the link between how much protein is made from the RNA is a whole nother disjunction, if you will. So what people like to see when they see, okay, the RNA is there, we know there's a virus present. Is the virus causing disease? A lot of viruses are asymptomatic, right? They're latent viruses, or they don't cause disease at all. They just happen to be passengers. So you really need to link it to other clinical conditions that would say, okay, we've got the virus and we have some sign of the virus doing bad things. And that's where it's really shined. It's also an absolute workhorse in genomics. Like, we use PCR all day long to sequence organisms and to amplify, small amounts of DNA out of a big soup of different DNA that we want to study. So it's an incredibly powerful technique. But using it in a vacuum, I think, is where it's gone wrong. Is everyone just said, we're in a state of panic. We're going to test everybody with PCR. And the reason it got used is because it's the only test we have that scales.
Yeah.
All these other things we're asking to be done are usually more complicated and more expensive. But PCR we can do in mass like a factory.
Yeah.
And so everyone resorted to that as a triage. The problem is we didn't understand the biology of the virus, and it didn't triage very well because the vast majority of people that it picked up weren't infectious. So it was a completely useless track and trace tool. If you pour 90% false positives into a track and trace system, it blows up. Because now you're 6 degrees away from Kevin Bacon turns into 1 degree, and everyone is guilty. Right. Because you start expanding the people who are positive into their family members. A bunch of them probably are non-infectious and are positive. They all have contact maps, and it's just the chain reaction is really the contact tracing system. So if you feed in low specificity data to contact tracing, it's worthless, which is what we saw. They didn't contact trace their way out of this. The pandemic came and went no matter what they did and how much money they blew. They couldn't slow it down. And if you look at the actual incident rate of this in countries that did testing versus countries that didn't do testing, you can't see a difference.
Yeah.
I mean, that's a sign from a macroeconomic standpoint that this isn't helping. We're just turning the wheels and greasing lobbying and greasing particular bottom lines. That doesn't mean PCR is invaluable. There's tremendous valuable asset we have here for measuring a lot of things about biology. But when it's used blindly as if it's a physician is when it goes off the rails like this.
Yeah. Yeah. And we see a lot of this, what you just said, we saw a lot of that demonstrated with respect to the mitigation measures that were employed, like mass mandates and lockdowns and all that. I mean, the virus came and went. You saw the spikes came and went, the waves came and went, no matter what we did. Yeah. And so it seems like this one size fits all approach with all of this, from testing to mitigation and so on and so forth, was misguided.
It's political rain dances. It was just to demonstrate that they were doing something. It was an action bias.
Yeah,
and the politicians could point to we're doing something, even if it's as worthless as a rain dance. Don't question it.
Yeah,
we have nothing else.
Has the CDC or the government publicly acknowleded these deficiencies with the PCR tests when used to try and diagnose COVID cases?
Well, they've at least put in a 90-day moratorium. Right. CDC. So that's a sign that they're admitting that this was flawed from the start. That they know that you can now be PCR positive that long, and you shouldn't crowd the system if you've been positive in 90 days before. That wasn't in place for the majority of the pandemic. It's now in place when we don't really need it because Omicron's ripping through, and it seems to be a nothing burger for most of the part. So yes, they're slowly admitting, I think, defeat as they can subtly. But these don't come out as like press releases. We were wrong. They just they shift the policy. Like they quietly shifted the policy on what they were sequencing when Omicron came out. They moved from 32 CT sequencing to below 28. That enriched the people being sequenced for having the highest viral load. Now that got deployed differentially on people who were vaccinated versus unvaccinated, because the unvaccinated people had to get tested more than the vaccinated people. So they were able to play these games with showing that, oh, look, the unvaccinated people are PCR positive more. Well, it's because you forced them to test, and you're actually made an exception for people who are vaccinated.
Right.
And they moved around these numbers at really opportune times to make the numbers look like a pandemic was still in place when it wasn't.
Yeah.
And I think that's still going on today. So
you mentioned earlier that there was that China had a more effective test at the beginning. What test was that?
So Gao put out a paper when they first found this in China. They wrote up a PCR assay and published it. And for some reason, the Drosten one ended up on the WHO and racing through Eurosurveillance. And from what I've gathered, the one from China didn't have the mistakes that Drosten had. Drosten designed his off of SARS-1, and because he didn't have access to the virus. This was like basically there were only 6 patients worldwide when he published this.
Mm-hmm.
So he couldn't get access to the virus. So he estimated what the primers should be based on SARS-1. And as a result of that, there's an erratum on his paper from someone who pointed out the fact that he has mistakes in his primers, that they're the wrong bases, and it's going to amplify SARS-1. They chalked that up to being SARS-1 isn't around anymore. Who cares? Except the paper just came out from Beijing showing that SARS-1 was found in 20% of flu patients in the Institute of Pasteur, and it would likely leaked out of a U.S. Genomics center, the Venture Institute. So when they're saying SARS-1 wasn't around anymore, and then papers come out showing, well, it was around, maybe it was a lab contamination, maybe it was a lab leak, we don't know, but you can't make this assumption that it's wrong. And if your primers can amplify SARS-1 and you think they also amplify SARS-2, well, what about OC23 and all the other coronaviruses? Are you checking those?
Right.
Their paper did some work to look at other viruses, but if you look at the footnotes in that, they show that they had four questionable PCR results out of 192. So that was a 2% error rate right out of the gate onto the WHO, used globally. 2% error rate when the population prevalence of the disease is less than 2% is a disaster. And you can plug that into Bayes' theorem and see that that's a train wreck. You can't use a test that already demonstrates out of the gate that it has a 2% error rate, and you don't have internal controls. You don't even give the people the advice as to what the CT score should be. There's really no strong SOP, and your primers have proven to be wrong. I mean, the whole thing was race it out the door and let's print some money.
And you mentioned he didn't even have access to the virus itself.
No, he synthesized DNA to emulate it to verify the test, and then collected some samples he got from other hospitals. But when they were designing the primers, they were probably designing those in December. Because if you look at the time scale, I think it came out January 19th.
Yeah.
So to turn around a PCR test, you're usually ordering primers a couple weeks before. So he was working on this in late December, probably had some primers in his hand that dealt with SARS-1 and threw those at the problem, and didn't update it to reflect the new sequencing that was coming out. Even in that paper, one of the primer sets that they designed 3. They didn't target a human gene. They targeted 3 viral elements. One of them, they self-admit in the papers doesn't work. And the other 2, we put a paper out showing they form primer dimers and have other errors in them, and they create a lot of false positives. The scientific community, well, I wouldn't say the whole community, shot it down, but we wrote this and then submitted it back to Eurosurveillance saying, we think you made a mistake and you should retract the paper. They did another closed review that took 2 months, didn't share the reviews, and came out saying, we policed ourselves and we think everything's fine. One of their reviewers, Stephen Buston, leaked out that he was one of the reviewers in a podcast and that he never looked at the primers. So the people that shot down our concerns are on record saying they never really looked at the problem and they just rubber stamped the rejection of what we were saying.
Mm-hmm.
And the Neurosurveillance has kind of flown them around to see the scientific community has rejected this concern over this. But I think what's actually happened in the marketplace is people aren't using that test as much. They've moved on to Thermo's tests and other people's tests that have built in a lot more of these safeguards against against the problems that were in the initial design. But that snowballed into what we have today, is everyone testing out all the time.
Yeah, I mean, but for that paper, is it possible we could have been relying on another test, like the one from China that you mentioned, or something else?
Yes, it's possible.
At the time. Go ahead.
Yeah, it's possible that other tests would have gotten stronger foothold and had a lower false positive rate, or the rat test may have gotten more tension. I mean, one thing that was really bizarre through the whole pandemic is lots of intelligent scientists in this field, Michael Mina being one of them, were pounding on the table as to why they weren't allowing home testing. We had all the tools to do home testing, but they insisted we all get in a line with a bunch of other sick people and get tested in some bottlenecks. That's a bad idea. Or testing in hospitals. That's where it spreads. Home testing would have meant you could test in the privacy of your home, and this wouldn't become a public outrage over who's in and who's out of the in-groups. But they held the brakes on those for 2 years, and only recently have they kind of allowed the home test to come out after the pandemic's over. I think there was a desire for control, and they knew if the testing was centralized in key laboratories and they could scrape the data from those laboratories into public health dashboards that they could edit and manipulate, they could control the messaging. But the moment they let home testing decentralize this, the message, it was over, and the people would figure out on their own who was sick and who wasn't. They wouldn't need the government.
Yeah.
So I think they put the kibosh on all the decentralized testing just to maintain control of the pandemic messaging.
I mean, it seems, just judging from all this, it seems, I wouldn't say it's negligent, the way the government handled its reliance on this one test, and then the public health response that was developed as a result of COVID case counts that were developed as a result of using this test. It seems like this isn't negligent. This is something
It's
worse than
grossly
that.
negligent, yeah.
Yeah.
And for Michelle Walensky to get up there and say the vaccinated can't spread it, I think that happened a year ago today.
Yeah.
Right?
Yeah.
They had the data as to what the PCR scores would be for the people who are vaccinated and unvaccinated. I mean, that had to be known early on because that was the tool that they were using to monitor the Pfizer trial, which was PCR positivity.
Right.
Right. So they must have known at a very early stage that these things were, that you were going to be PCR positive even if you got vaccinated, and that posed a real problem to the logic of their vaccine passports. But they pushed ahead with them anyways, until enough public outrage showed up and enough papers showed up that the vaccinated sometimes had even higher viral loads than the unvaccinated. And they finally had a cave on that point after they'd done all the damage of mandating these things in various jurisdictions.
I think we had. I mean, we also knew we had the data coming out of Italy at the time, early 2021, January, February, where, I mean, the elderly were dropping like flies, obviously, and we knew that this was a pandemic where only a select portion of the population was at risk. And so I think we had all this information already, and we knew that we didn't need a one size fits all test to scale up to push everyone towards. I feel like we could have taken a more focused approach in medically diagnosing who had this virus and who didn't, based on the data that was coming out of Italy at the time, because some governors in this country relied on that data in a beneficial way, and others didn't, and just adopted these one size fits all test approaches, mitigation approaches, and so on.
Yeah, you're right on. I mean, the comorbidities alone are probably a better signature for who's at risk than, per se, the RNA test. You apply the RNA test to everyone in the population, vast majority of them, if they're positive, aren't at risk, kids included. But if you look at the comorbidities, those can bring up your risks, and your age in particular, those can bring up your risk 1,000-fold.
Yeah.
So there was never a more opportune time to do a very focused protection on this, because we knew early on that age was the largest contributor to this, and comorbidities from COPD, diabetes, cancer, and obesity. Those four things were triggers to escalate your risk substantially.
Right.
But RNA positivity, without any of those considerations, you're blind. You really can't predict. The vast majority of people who will be RNA positive won't give you clinical guidance. If you know their age, if they're diabetic, or any of these comorbidities, you have a much better sense of who's at risk than you do from a general RNA test with none of that information.
This is, I mean, it's harrowing talking about all this and just the way it all played out. It just seems so irresponsible. But we've gone about an hour. I really appreciate your time and the information you provided. It's really interesting. It's really informative and educational. And I think that there's a comeuppance coming where people are going to realize that many of these approaches were misguided and ill-advised, and we could have done it differently. And those alternative approaches could have impacted our economy and people in a much more positive way than what we did.
Well, I think it's important for your audience in particular to hear this, because this is old saying, like, never attribute to malice what can just be ignorance, right? I don't think that's what's going on here. I think these people truly knew that they were going to cause harm, but they did it anyway for revenues, for vaccine interests, for political interests, and we can't let them get away with it. And we need to structure something in the law that prevents this, because many of them did this by pushing the boundaries of the law, knowing that it's going to be very hard to prosecute.
Yeah, I agree. I've come right up against that. Many of these government officials, especially at the local and state level, just did whatever they wanted. They don't care. People ask me, how can they do that? But the point is that they do it, and they know that they can likely get away with it. And few people are going to challenge it. The ones who don't, the ones who do challenge it, then you run up against the court system, which you've now seen, which is very resistant to chipping away at this stuff or to calling into question these measures, because they traditionally like to defer to executive power and the executive decision making, because they're the ones with the information. They're the ones that are closest to these situations. But at the end of the day, I mean, the PCR test is a classic example of a totally misguided approach where they had information indicating that they could have adopted something different that would have worked a lot better. And we had this information at the time. And this is not only it's negligent, grossly negligent. I think it was intentional, as you just said. It's truly it's disheartening to see this stuff.
They very much could have done it differently, even with PCR. They could have tested people twice.
Yeah.
If you see your CT score change on day 1 to day 2, like the viral load goes up on day 2, you're on the front end of the infection and you're getting worse and you're probably infectious and you should hide in quarantine. If you see it go the other direction, you're on the back end of the curve and you're probably no longer infectious. So they could have simply just gone to 2 tests and solved this problem. There's other technology out there that does this live dead thing, needs a little bit more validation, but people have been looking at ways where they only amplify viral particles that they know are infectious. There's papers on this, right? None of that got implemented. And you have to ask, like, why? It was obvious that this was the problem. How come none of this made it into practice? And you're like, well, doubling the amount of PCR could be a problem. Bullshit. Like they increased the rate of PCR over COVID 100-fold in like a year.
Right.
They had plenty of capacity to do a 2-time point test to understand who was infectious and who wasn't, so they didn't wreck the economy with over quarantine. But they didn't do that. And it's because I think they wanted to use these quarantines as a political lever to drive division in society, so people would fight amongst themselves and not fight with how much money they're printing in government. And so I do think it's very much, it's not just ignorance. It's actually like they're hiding under the banner of ignorance with malice.
Yeah.
And we can't let them get away with it, because they'll do it again and again and again every time they find a chance to do it. I
agree. Well, thank you again. I really appreciate your time. Very informative, and love to have you back at some point. I know we need to talk about the vaccines at some point, and because there's a lot there as well.
Yeah.
But I think this topic itself was meaty enough that we could cover. We could devote a whole hour to it. So again, thank you very much. I really appreciate your time, Kevin.
Okay. Awesome. All right. Take care.